Today we will be covering scientific inquiry in DNA. Scientific inquiry. Scientific inquiry is the diverse ways in which scientists study the natural world and propose explanations based on the evidence derived from their work and experiments. Scientific inquiry of the structure of DNA has been an ongoing process from the time of Gregor Mendel with his pea plant genetics experiment and Thomas Hunt Morgan, who discovered that genes exist as a part of chromosomes. After their experiments, other scientists made new discoveries, adding on to Mendel and Morgan's information and experimental data. In summation, Mendel's heritable factors and Morgan's genes on chromosomes are composed of DNA. This was further proven by all the scientific experiments that followed. This also answers question 1 on your handouts. What did Morgan and Mendel's experiments have in common? Both proved that Mendel's heritable factors and Morgan's genes on chromosomes are composed of DNA.
In 1928, Frederick Griffith did an experiment to test whether bacteria were capable of transferring genetic information through a process called transformation. Since pneumonia was a serious problem after World War I, Griffith decided to study the possibility of creating a vaccine. Frederick Griffith used 2 strains of pneumococcus bacteria which infect mice. One type was smooth and one type was rough. The smooth strain covered itself with a polysaccharide capsule that protected it from the host's immune system. Therefore, the mouse died. The rough strain did not have this polysaccharide capsule. Therefore, the immune system was able to fight and kill off the bacteria. That's the answer to question 1 in your handout. What was the main difference between the 2 strains of pneumococcus bacteria that Frederick Griffith used? The answer is the smooth strain covered itself with a polysaccharide capsule, while the rough strain did not. Then he tested killing the smooth strain with heat. This killed the smooth strain of bacteria, so the mouse lived. That's the answer to question 2. How did Frederick Griffith try to kill the smooth strain, and did it work? The answer is yes, it worked. He killed the smooth strain with heat. Finally, once the smooth strain was killed by heat, Griffith added the remnants to the rough strain in this experiment. While neither alone killed the mouse, both combined did kill the host. Frederick Griffith was then able to isolate both the live, smooth, and rough strains of pneumococcus from the blood of the dead mice. He concluded that the rough strain had transformed into the smooth strain, forming a polysaccharide capsule, because the remnants of the smooth strain had somehow told them to. That's the answer to question 3 in your handout. What was Griffith's conclusion? His conclusion was that the rough strain had transformed into the smooth strain. He called this the transforming principle. Today we know that this transforming principle was the DNA of the smooth strain bacteria. This is the answer to question 4 in your handout. What do we know today that the transforming principle is? We know the transforming principle is DNA. While the bacteria of the smooth strain had been killed, the DNA had survived and was taken up by the rough strain bacteria. The DNA from the smooth strain coded for the polysaccharide capsule, which protected the bacteria from the immune system. The exact nature of the transforming principle, DNA, was later verified by experiments done by Avery, MacLeod, and McCarty, and by Hershey and Chase. Griffith's work set the stage for Oswald Avery's 14-year effort to identify what exactly was the transforming substance, which was DNA.
Oswald Avery. Oswald Avery's experiment was about finding the transforming principle. He used a series of experiments using strains of Pneumococcus bacteria which cause pneumonia. This is the answer to number 1. What is Avery's transforming principle experiment? Pneumococcus grows in the body of the host, but can also be grown on solid or liquid cultures. This is the answer to number 2 in the blanks. Pneumococcus grows in the body of the host, but can also be grown on solid or liquid cultures. Oswald Avery read about Frederick Griffith's experiments and became very interested in the transforming principle. He began experimenting using a test tube assay instead of mice, which is what Frederick Griffiths used. This is the answer to number 3. Whose experiments inspired Avery's experiments? That would be Frederick Griffiths. His experimental procedure. First, he used detergent to lyse the heat-killed S cells, then used lysate for transformation assays. The test tube assays showed that the heat-killed S lysate could change R to S, and then this told us that something in the lysate was the transforming principle. To find the transforming principle, they incubated the heat-killed S lysate with an enzyme, S3, which chewed up the sugar coat. He tested the transforming ability of the sugar coatless S lysates, and it was still able to transform. So then this told us that the R strain was not just assembling a new S sugar coat from the pieces. Next, he incubated the coatless S extract with protein-digesting enzymes, trypsin and chymotrypsin. This is the answer to number 4 on the handout. What were the protein-digesting enzymes used to incubate the coatless S extract? That would be trypsin and chymotrypsin. He then tested these lysates' ability to transform, and it was still able to. So this means the transforming principle was not the protein. While testing and purifying the lysate, he precipitated the nucleic acids DNA and RNA with alcohol. He was the first to isolate nucleic acids from Pneumococcus. This is the answer to number 6. This is the answer to number 5. What did he use to precipitate the nucleic acids, DNA, and RNA? He used alcohol. For number 6, so far the transforming principle was not the sugar coat or the protein, so he predicted it would be one of the nucleic acids. He tested the transforming ability of the solution by dissolving the precipitate in water. He destroyed the RNA using the RNase enzyme, and then he tested this solution for its ability to transform. What was left was pure DNA. For the final test, he incubated the solution with the DNA-digesting enzyme DNase. Then he tested for the transforming ability, and it was unable to transform. He and his colleagues concluded that DNA was the transforming principle. These results were published in 1944. This is the answer to number 7. What did Oswald Avery and his colleagues conclude in 1944? DNA is the transforming principle. This is just an overview of his entire experiment.
Rosalind Franklin and Maurice Wilkins. Rosalind Franklin and Maurice Wilkins were two colleagues who worked together at the King's College in London in 1951. That's question 1 on your handout. Franklin and Wilkins worked at the King's College in London in 1951. They were hired to improve the school's use of X-ray crystallography. While they were there, they used X-ray crystallography to determine the DNA double helix shape. That's question 2. The answer is X-ray crystallography. What can X-ray crystallography provide for us? In X-ray crystallography, the diffraction patterns created can provide a lot of information about the shape and structure of a molecule. That's question 3, the shape and structure of a molecule. The process of X-ray crystallography works like this. If a stream of X-rays is directed at a crystallized substance, some rays are diffracted, or scattered, as they encounter the atoms. The scattered X-rays then interfere with one another and produce spots of different intensities, and these can be recorded on photographic film. This is the answer to question 4, which asks basically about the process of X-ray crystallography. Question 5 asks what Wilkins and Franklin's X-ray photographs show about DNA structure. Specifically, the data in the photo showed that the DNA was a 2-stranded helical shape. This photo is a product of Franklin and Wilkins X-ray crystallography. It is taken of DNA. The X shape in the middle indicates that the DNA has a circular or helical shape. It also showed that DNA was actually 2-stranded, and not three-stranded or one-stranded, as previously hypothesized. From this information, Franklin was able to calculate the basic dimensions of the DNA model. This answers your last question, question 6. The information Franklin was able to derive from the model was that she was able to calculate the basic dimensions of the DNA. Watson and Crick are most credited with the findings of DNA structure, although they received much of their information from Franklin and Wilkins. In 1962, Maurice Wilkins, Watson, and Crick won the Nobel Prize in Physiology or Medicine. Although Franklin had already passed away and was not able to win, she was not even nominated for the prize.
In 1952, Hershey and Chase performed their famous blender experiment. This is the answer to question 1 on your handout. What experiment took place in 1952? The blender experiment. The pair set out to show that DNA is the genetic material of a phage called T2. A phage is a virus that infects bacteria. This is question 2 on your handout. What is a phage? They planned an experiment showing that one of the two components of T2, either DNA or protein, enters an E. coli cell during infection. This is question 2. What was Hershey and Chase's experiment designed to show? They designed an experiment to show that one of the two components of T2, either DNA or protein, enters an E. coli cell during infection. This is a picture of multiple phages infecting a bacterial cell. Hershey and Chase used radioactive sulfur and phosphorus to trace the fates of protein and DNA of T2 phages that infected bacterial cells. This is number 4 on your handout. What two elements did Hershey and Chase use to trace DNA and protein? And that was radioactive sulfur and phosphorus. Their wish was to see which of these molecules entered the cells, and they planned to reprogram them to make more phages. This is the written out version of their experiment, which is the answer to question 5 on your handout. Briefly explain Hershey and Chase's experiment, but I'm going to use a picture to explain it. Hershey and Chase used 2 batches of protein. In the first batch, phages were grown with radioactive sulfur and then incorporated into the pink phage protein. Phages in the second batch were grown with radioactive phosphorus, which was then incorporated into the blue phage DNA. First, they mixed radioactively labeled phages with bacteria. The phages infected the bacterial cells. Next, they agitated the mixture in a blender to free phage parts outside the bacteria from the cells. Then they centrifuged the mixture so that the bacteria formed a pellet at the bottom of the test tube. The lighter free phages and phage parts remained suspended in the liquid. They then measured the radioactivity of the pellet and the liquid. In the first batch of radioactive sulfur phages, the radioactivity was in the liquid. In the second batch of phages, the radioactivity was found in the pellet. Bacterial cells with radioactive phage DNA released new phages with some radioactive phosphorus. Hershey and Chase concluded that phage DNA entered bacterial cells, but phage proteins did not. Thus, DNA, not protein, functions as the genetic material of phage. This is the answer to question 6 on your handout. Explain how the results of the blender experiment prove that DNA is the genetic material of a phage. The answer is that in the first batch of radioactive sulfur phages, the radioactivity was in the liquid. In the second batch of phages, the radioactivity was found in the pellet. So Hershey and Chase concluded that phage DNA entered bacterial cells, but phage protein did not. Again, DNA, not protein, functions as the genetic material of phages. In 1969, Hershey and Chase won the Nobel Prize in Physiology or Medicine for their discovery that DNA is the genetic material of phages.
Perhaps the most famous scientists known for working on DNA is James Watson and Francis Crick. They are responsible for giving us the model of DNA that we still use today. Watson had gone to Cambridge University to visit the laboratory of Maurice Wilkins, when he saw an X-ray diffraction image of DNA produced by Rosalind Franklin. Watson was already familiar with the type of X-ray diffraction pattern that helical models produce. Therefore, after seeing this picture, Watson concluded that DNA was helical in shape. The pattern in the photo also implied that it was made up of 2 strands, instead of 3 as previously suggested. Watson and Crick then began to build models that conformed to what they already knew about DNA. They came up with this model. They knew that the sugar phosphate backbones must be on the outside, according to Rosalind Franklin's work. Using logic, they deduced that nitrogenous bases should be in the middle, these things right here, because of their relatively hydrophobic nature. Being on the inside of the DNA structure would shield them from the surrounding aqueous solutions. Likewise, the negatively charged phosphate groups would be on the outside. It's these things right here. Yeah, these. Where they would be far enough apart to not repel each other. In this model, the 2 sugar phosphate backbones are anti-parallel, meaning their subunits run in opposite directions. Let's pause and look at your handout. Question number 1 says, true or false, nitrogenous bases are located on the inside of the DNA structure. The answer is true. Nitrogenous bases are on the inside, as seen on this model, and the sugar phosphate backbone is on the outside. Question number 2 is the follow-up question. Why must the nitrogenous bases be on the inside of the DNA structure? This is because they are hydrophobic, and they must get away from the aqueous solution surrounding the DNA model. Okay, moving on. The nitrogenous bases of the double helix are paired in specific combinations: adenine with thymine and guanine with cytosine. Watson and Crick came to this conclusion by trial and error, which doesn't sound like the most scientific process, but sometimes it's all that you can do. The diameter of the double helix was already known from the X-rays. It was then up to Watson and Crick to find out how the nitrogenous bases lined up. They first paired adenine with adenine and cytosine with cytosine, aka a purine and a purine and a pyrimidine and a pyrimidine. This, however, did not fit the required spacing, which is the red dotted line. Therefore, a purine must be paired with a pyrimidine because it's the only thing that fills up the correct spacing. According to Chargaff's second rule, Chargaff's, however you say his name, the number of A and T bases and the number of C and G bases are equal. Therefore, Watson and Crick concluded that A always pairs with T and C always pairs with G. Let's pause again and answer the remaining questions on your handout. You can go back up to me now. How did Watson and Crick know that a purine must be paired with a pyrimidine? X-ray pictures showed that the diameter of the helix and the only combination that built this space was a purine with a pyrimidine. Fourth question. Which of the following will be paired up according with Watson and Crick? A. Adenine and guanine. B. Adenine and cytosine. C. Cytosine and guanine. Or D. Cytosine and thymine. The correct answer is C. Cytosine and guanine. The 2 scientists published all this information in April of 1953 in a 1-page paper in the journal Nature. The double helix has since become the symbol of molecular biology. Watson and Crick, along with Maurice Wilkins, were awarded the Nobel Prize in 1962 for their work. So in conclusion, many scientists contributed to the current model of DNA. Gregor Mendel discovered heritable factors. Thomas Hunt Morgan showed us that genes exist as part of chromosomes. Frederick Griffith found a chemical substance that was able to transform the function of a cell. Oswald Avery then concluded that DNA was the transforming agent. Alfred Hershey and Martha Chase confirmed that DNA functions as the genetic material. Rosalind Franklin and Maurice Wilkins also discovered many attributes of DNA that led to James Watson and Francis Crick's famous DNA model. All of this would not have been possible without the process of scientific inquiry.